Welcome to the second lecture of Physiology. In this lecture, we will discuss about the organelles in cytoplasm. Cytoplasmic organelles are the cellular structure embedded in the cytoplasm. Organelles are considered as small organs of the cell. Some organelles are bound by limiting membrane and others do not have limiting membrane. Each organelle is having a definite structure and specific function. Organelles with limiting membrane. Endoplasmic reticulum. Endoplasmic reticulum is a network of tubular and microsomal vesicular structures. It is covered by a limiting membrane. The lumen of endoplasmic reticulum contains a fluid medium called endoplasmic matrix. The endoplasmic reticulum forms the link between nucleus and cell membrane. Types of endoplasmic reticulum. There are two types of endoplasmic reticulum: rough endoplasmic reticulum, smooth endoplasmic reticulum. Rough endoplasmic reticulum. Rough appearance is due to attachment of granular ribosomes to its outer surface. Hence, it is also called the granular endoplasmic reticulum. Rough endoplasmic reticulum is vesicular or tubular in structure. Function of rough endoplasmic reticulum. First function of rough endoplasmic reticulum is synthesis of protein. It is concerned with the synthesis of protein in the cell. It is involved with the synthesis of mainly those proteins which are secreted from the cells, such as insulin from beta cells of islets of Langerhans in pancreas and antibodies from B lymphocytes. Ribosomes arrange the amino acids into small units of protein and transport them into the rough endoplasmic reticulum. Here the carbohydrates are added to the protein units, forming the glycosylated proteins or glycoproteins, which are arranged in the form of reticular vesicles. These vesicles are transported mainly to Golgi apparatus for further modification and processing. Few vesicles are transported to other cytoplasmic organelles. The second function of rough endoplasmic reticulum is degradation of worn-out organelles. Rough endoplasmic reticulum also play an important role in the degradation of worn-out cytoplasmic organelles like mitochondria. It wraps itself around the worn-out organelles and forms a vacuole, which is often called the autophagosomes. Autophagosomes is designed by lysosomal enzymes. Smooth endoplasmic reticulum. It is the endoplasmic reticulum with smooth appearance. It is also called agranular reticulum. It is formed by many interconnected tubules. So it is also called tubular endoplasmic reticulum. Function of smooth endoplasmic reticulum. The first function of smooth endoplasmic reticulum is synthesis of non-protein substances. Smooth endoplasmic reticulum is responsible for synthesis of non-protein substances such as cholesterol and steroids. This type of endoplasmic reticulum is abundant in cells that are involved in the synthesis of lipids, phospholipids, lipoprotein substances, steroid hormones, and sebum. In most of the other cells, smooth endoplasmic reticulum is less extensive than the rough endoplasmic reticulum. The second function of smooth endoplasmic reticulum is role in the cellular metabolism. Outer surface of smooth endoplasmic reticulum contains many enzymes which are involved in various metabolic processes of the cells. Storage and metabolism of calcium is also the function of smooth endoplasmic reticulum. Smooth endoplasmic reticulum is the major site of storage and metabolism of calcium. In skeletal muscle fibers, it releases calcium, which is necessary to trigger the muscle contraction. Catabolism and detoxification. Smooth endoplasmic reticulum is also concerned with the catabolism and detoxification of toxic substances like some drugs and carcinogens in the liver. Golgi apparatus. Golgi apparatus or Golgi body or Golgi complex is a membrane-bound organelle involved in the processing of proteins. Each cell has one Golgi apparatus. Some of the cells may have more than one Golgi apparatus. Golgi apparatus consist of 5 to 8 flattened membranous sacs called the cisternae. Function of Golgi apparatus. Major functions of Golgi apparatus are processing, packing, labeling, and delivery of proteins and other molecules like lipids to different parts of the cell. Processing of material. Vesicle contain the glycoproteins and lipids are transported into Golgi apparatus. Here the glycoproteins and lipids are modified and processed. The second function of Golgi apparatus is packaging of materials. All the processed materials are packed in the form of secretory granules, secretory vesicles, and lysosomes, which are transported either out of the cell or to another part of the cell. Because of this, Golgi apparatus is called the post office of the cell. Labeling and delivery of the material. Finally, the Golgi apparatus sorts out the processed and packed material and labels them, such as phosphate group, depending upon the chemical content for delivery to their proper destination. Hence, the Golgi apparatus is called shipping department of the cell. Lysosomes. Lysosomes are the membrane-bound vesicular organelles found throughout the cytoplasm. The lysosomes are formed by Golgi apparatus. The enzymes synthesized in rough endoplasmic reticulum are processed and packed in the form of small vesicles in the Golgi apparatus. Types of lysosomes. There are two types of lysosomes. Primary lysosomes, which are pinched off from Golgi apparatus. It is inactive in spite of having hydrolytic enzymes. Secondary lysosomes, which is active lysosome. It is formed by the fusion of primary lysosome with phagosome or endosome. Function of lysosome. Lysosomes are often called carbide system of the cell because of their degradation activity. About 50% hydrolytic enzymes known as acid hydroxylases are present in the lysosomes, through which lysosomes execute their functions. Importance of lysosomal enzymes. Proteases, which hydrolyze the proteins into amino acid. Lipases, which hydrolyze the lipids into fatty acids and glycerides. Amylases, which hydrolyze the polysaccharides into glucose. Nucleases, which hydrolyze the nucleic acid into mononucleotides. Mechanism of lysosomal function. Heterophagy. Digestion of extracellular materials engulfed by the cell via endocytosis. Autophagy. Digestion of intracellular materials such as worn out cytoplasmic organelles. Now we discuss the specific function of lysosomes. The first function of lysosomes is degradation of macromolecules. Macromolecules are engulfed by the cell by means of endocytosis. The macromolecules such as bacteria engulfed by the cell via phagocytosis are called phagosomes or vacuoles. The other macromolecules taken inside via pinocytosis or receptor-mediated endocytosis are called endosomes. The primary lysosomes fuses with the phagosomes or endosomes to form the secondary lysosomes. The pH in the secondary lysosomes becomes acidic and the lysosomal enzymes are activated. The bacteria and the other macromolecules are digested and degraded by these enzymes. The secondary lysosome containing these degraded waste products moves through cytoplasm and fuses with the cell membrane. Now the waste products are eliminated by exocytosis. The second function of lysosome is degradation of worn out organelles. The rough endoplasmic reticulum wraps itself around the worn out organelles like mitochondria and form the vacuoles called autophagosomes. One primary lysosome fuses with one autophagosome to form the secondary lysosome. The enzymes in the secondary lysosomes are activated. Now these enzymes digested the contents of autophagosomes. Removal of excess secretory products in the cell. Lysosomes in the cells of secretory glands remove the excess secretory products by degrading the secretory granules. Secretory function. Recently, lysosomes having secretory function called secretory lysosomes are found in the sum of the cells, particularly in the cell of immune system. The conventional lysosomes are modified into secretory lysosomes by combining with the secretory granules. Peroxisomes. Peroxisomes or microbodies are the membrane-limited vesicles like the lysosomes. Unlike lysosome, peroxisomes are pinched off from endoplasmic reticulum and not from the Golgi apparatus. Peroxisome contains some oxidative enzymes such as catalase, urate oxidase, and D-amino acid oxidase. Function of peroxisomes: break down the fatty acid by means of process called beta oxidation, degrade the toxic substances such as hydrogen peroxide and other metabolic products by means of detoxification, form the major site of oxygen utilization in the cells, accelerate the gluconeogenesis from fats, degrade purine to uric acid, participate in the formation of myelin, play a role in the formation of bile acid, centromere and centrioles. Centromere is the membrane-bound cellular organelle situated almost in the center of the cell, close to nucleus. It consists of two cylindrical structures called centrioles, which are made up of the protein. Centrioles are responsible for the movement of chromosome during the cell division. Secretory vesicles. Secretory vesicles are organelles with limiting membrane and contain the secretory substances. These vesicles are formed in the endoplasmic reticulum, are processed and packed in the Golgi apparatus. Mitochondria. Mitochondria is a membrane-bound cytoplasmic organelle concerned with producing the energy. It is a rod-shaped or oval-shaped structure. It is covered by a bilayered membrane. The outer membrane is smooth and encloses the content of mitochondria. Function of mitochondria. The first function of mitochondria is production of energy. Mitochondria is called the powerhouse or power plant of the cell because it produces the energy required for cellular functions. The energy is produced during the oxidation of digested food particles like proteins, carbohydrates, and lipids by the oxidative enzymes in cristae. During the oxidative process, water and carbon dioxide are produced with release of energy. The released energy is stored in mitochondria and used later for the synthesis of ATP. Synthesis of ATP. The components of respiratory chain in mitochondria are responsible for the synthesis of ATP by utilizing the energy by oxidative phosphorylation. ATP molecules diffuse throughout the cell from mitochondria. Whenever energy is needed for cellular activity, the ATP molecules are broken down. Apoptosis. Cytochrome C and second mitochondria-derived activator of capsaicin secreted in mitochondria are involved in the apoptosis. The other function of mitochondria includes storage of calcium and detoxification of ammonia in liver. Organelles without limiting membrane. Ribosomes. Ribosomes are the organelles without limiting membrane. These organelles are granular and small dot-like structures with the diameter of 15 nanometer. Ribosomes are made up of 35% of protein and 65% of ribonucleic acid. RNA present in ribosomes is called ribosomal RNA. Types of ribosomes. There are two types of ribosomes. Ribosomes that are attached to rough endoplasmic reticulum and free ribosomes that are distributed in the cytoplasm. Function of ribosomes. Ribosomes are called protein factories because of their role in the synthesis of proteins. Messenger RNA carries the genetic code for protein synthesis from nucleus to the ribosomes. Cytoskeleton. Cytoskeleton is a cellular organelle present throughout the cytoplasm. It determines the shape of the cell and gives support to the cell. Cytoskeleton consists of three major protein components: microtubules, intermediate filaments, and microfilaments. Microtubules. Microtubules are the straight, hollow and tubular structure of the cytoskeleton. These organelles without the limiting membrane are arranged in different bundles. Structurally, the microtubules are formed by the bundle of globular protein called tubulin. Intermediate filaments. Intermediate filaments are the structure that form a network around the nucleus and extend to the periphery of the cell. The intermediate filaments are formed by rope-like polymers which are made up of fibrous proteins. Microfilaments. Microfilaments are long and fine thread-like structures. These filaments are made up of non-tubular contractile proteins called actin and myosin. Actin is more abundant than myosin. Thank you.